Viruses. Incredibly diverse, capable of infecting all domains of life. They hijack cells, turning them into replication factories to make billions of copies of themselves. What's their overall goal? To transmit. One cell to another, one individual to another, one community to another. They cross borders, they can jump species, they are able to mutate and adapt, capable of all these things, yet they are barely classified as living. They are on the edges of life. Let's talk about viruses. The common cold, influenza, COVID-19. These are all infections caused by viruses, a submicroscopic infectious agent. Most viruses are so small they cannot be viewed under a light microscope, rather require an electron microscope for visualization. They come in different shapes, but share a similar structure. A viral particle is known as a virion. A virion consists of a protective protein coat called a capsid, usually made up of several different alternating proteins. This protein coat encapsulates a nucleic acid genome. Some viruses also have an extra lipid outer layer known as a viral envelope, which is derived from the host cell membrane. So, how do these viral particles infect cells? Let's look at an example. This is a Picornavirus, a family which includes the causative agents of the common cold. Pico meaning small and ana meaning RNA, a small RNA virus. Picornavirus capsids consist of 4 proteins arranged as a pentamer. 3 of these proteins are exposed on the surface. Between the pentameric structures, there are deep canyons where the receptors on the host cell can insert. The shape of the canyons determine which receptor the virus binds to. The virion attaches to a receptor on the cell surface. This can trigger an uptake of the virus. Binding to the receptor also triggers a shift in the viral structural proteins, forming a pore, allowing the RNA genome to be released into the cell. This RNA encodes for multiple proteins, including structural proteins that form the capsid, as well as non-structural proteins. The RNA is translated by the ribosomes in the host cell, producing both structural and non-structural proteins. The non-structural proteins recruit cellular membranes to form a viral factory, where the original viral RNA is encapsulated. Another non-structural protein that was produced is called RNA-dependent RNA polymerase, which uses the original viral RNA strand as a template to transcribe it into negative sense RNA and subsequently multiple positive sense RNA, which are identical to the original viral RNA. The new positive sense RNA can exit the viral factory and be translated by ribosomes to produce more structural and non-structural proteins. The virion is then assembled, with the structural proteins forming the capsid encapsulating a positive sense RNA. This process continues until thousands of virions are produced, too much for the cell to handle. The cell lyses open, releasing the virions, which are then capable of infecting more cells. What happens next? The virus is infecting your cells. They are dying as more virions are being released. Cell death releases signals that recruit immune cells, like macrophages, lymphocytes, and neutrophils. They all migrate to the site of infection. They release chemical mediators that cause a cascade of events resulting in cold symptoms, such as sneezing, mucus production, fever, and coughing. This is all a response initiated by the immune system to try and clear the virus. However, This is also how the virus escapes and transmits to the next individual. Influenza A is another virus which infects humans. However, it has its origins from aquatic birds. People often catch influenza A seasonally, which is called the common flu. However, occasionally, there are strains of influenza A that can spread across the world, known as a pandemic. The most recent influenza pandemic was in 2009, known as swine flu. So, why do some strains of influenza become pandemic while others don't? Let's explore this further. Influenza is classified by their HA or hemagglutinin and NA or neuraminidase antigenic genotypes. These are important proteins that the virus uses to infect cells. There are also 6 other genome sequences encoding important viral proteins, so 8 in total, including HA and NA. You may be familiar with the swine flu genotype, H1N1, hemagglutinin type 1, and neuraminidase type 1. There are currently 18 different hemagglutinin genotypes and 11 different neuraminidase genotypes, as well as other types for the other 6 genome sequences. These different genotypes, for example H2N2, circulate in different species. However, occasionally they can jump species. Suppose an individual who was infected with H1N1 influenza virus was also the recipient of the H2N2, which jumped species from a bird to a human. The 2 different influenza viruses can reassort its 8 genome segments in that individual, resulting in a new progeny virus with genome segments from both parent viruses. This is known as genetic shift and may lead to a pandemic. One genome segment which is crucial for reassortment to cause a pandemic is hemagglutinin. This is because hemagglutinin is the main molecule which neutralizing antibodies are produced. For example, this individual has had H1N1 virus before and contains antibodies against it. They are neutralizing the virus, targeting them for destruction. However, H2N2 has jumped species into the individual and has reassorted with H1N1 to produce H2N1. This individual's immune system has never encountered H2 before, and therefore the individual is not protected, and neither is anyone else. It spreads throughout the population, from one community to another, crossing borders leading to mass morbidity and mortality. It is now classified as a pandemic. Many years later, after the initial outbreak, H2N1 has circulated around the world. Most people have antibodies against it, which will neutralize the virus quickly. However, the virus has a trick up its sleeve. It is able to mutate. It is still H2N1, however, it has mutated the sites on its H2 molecule that the antibodies recognize. This is known as genetic drift and is the primary reason why we get seasonal flus and require a new influenza vaccine every few years. So, we have lost immunity because of genetic drift. Does that mean seasonal flus are just as deadly as the initial pandemic outbreak? Well, no. Viruses mutate all the time. Deadly viruses, which initially have a high fatality rate, usually trend towards being less deadly, or in other words, they have attenuated. Seasonal flus are nowhere near as deadly as the initial pandemic outbreak. This is explained by natural selection. Let's explore this idea through another example, this time in rabbits. Myxoma virus is transmitted through mosquitoes or fleas. They reside in the mouthparts of the insect. After probing their mouthpart into another animal, the virus is wiped off and transmitted. Myxoma virus's natural host is the Brazilian cottontail rabbit, causing cutaneous fibromas, which is essentially non-lethal lumps on the skin. Myxoma virus is also capable of infecting European rabbits, causing the disease known as myxomatosis, with symptoms of facial and body swelling resulting in death weeks after infection. Not only is it spread through mosquitoes in European rabbits, but also infected rabbits are capable of transmitting the virus to others by contact. So why is there a difference in the response and the lethality between the two species? Well, the Brazilian cottontail rabbit has evolved with Myxoma virus in an arms race. Myxoma virus has over 40 genes that have potential immunosuppressive effects. These virulence factors evolved to keep the Brazilian cottontail rabbit's immune system at bay for long enough for the virus to infect, replicate, and transmit, resulting in non-lethal symptoms. European rabbits, on the other hand, have not evolved alongside Myxoma virus, and therefore the same proteins encoded by these genes overwhelm the host's immune system, resulting in death. European rabbits are considered pests in certain countries, such as Australia. Introduced during European settlement, they have spread across the country. In the 1950s, an experiment was conducted using Myxoma virus as a biological control to eliminate European rabbit pests from Australia. If you remember before, Myxoma virus is spread through mosquitoes. So, the big brains in Australia released the virus in a mosquito-rich area during autumn and winter. However, the virus wasn't spread because those mosquitoes did not feed on the rabbits. It wasn't until the summer, after rivers flooded, did the virus take hold. Massive outbreaks of myxomatosis occurred in the floodplains, spread through close contact. This experimental release was so successful, they proceeded with large-scale releases. The strain of virus was very lethal initially, killing 99.8% of infected rabbits, with an average survival time of 10.5 days. 10 years later, however, the virus has become less effective as a biological control agent, with more rabbits capable of surviving. So how did this happen? One half can be explained by the rabbits. The virus initially killed 99.8% of infected rabbits. The other 0.2% survived, recovered, and are now immune. They replicate, passing on their immunity to their offspring. So over the 10 years, we have one population of rabbits that have inherited the immunity. Next, let's go to the virus. Initially, the virus was very effective at killing rabbits. This is where the virus faces problems. When there are no rabbits left, the virus cannot be transmitted and therefore will die out. Viruses mutate, and it so happens that those viral strains that mutated to become less lethal were more likely to transmit and persist in the environment compared to those strains that were more lethal. So, over the 10 years, the virus has become more attenuated, meaning that a higher proportion of the rabbits infected with the virus will survive, recover, and become immune. So, we have two driving forces: higher immunity from the rabbits and a selection for lower lethality for the virus, meaning that over time it is nowhere near as deadly as the initial outbreak. Viruses are capable of spreading across the world, initially spreading from one cell to another, then other individuals. More individuals become susceptible as viruses rearrange and mutate. However, viruses walk a fine line between virulence and transmission. They must find a balance between the two in order to be successful. Their ability to adapt mimics the dynamics of living organisms, yet viruses are barely considered alive. They are truly on the edges of life.